This video is about the experiment 7 having aim to perform the microbiological assay of given antibiotics by cup plate method and disc plate method. References. Requirement. Antibiotics. Antibiotic disc, nutrient agar media, Petri plates, conical flask, autoclave, borer, theory. Microbiological assay. The inhibition of microbial growth under the standard condition may be utilized for demonstrating the therapeutic efficiency of an antibiotic. Any subsequent change in the antibiotic molecule which may not be detected by chemical methods can be revealed by microbial activity. Here, microbial assays are very useful to know the possible loss of antibiotic activity. They are also routinely employed to determine the potency of all antibiotic preparations at various stages of development from the crude forms to finished product. The microbial assay is based upon the comparison of inhibition of bacterial growth by measured amount of antibiotic to be examined with that produced by the known concentration of standard preparation of the antibiotic having known activity. Procedure. Preparation of media. A. Prepare nutrient agar or Mueller-Hinton agar according to the manufacturer's instructions. B. Sterilize the media by autoclaving at 121 degrees Celsius for 15 minutes. C. Pour the sterilized agar into sterile Petri dishes to a uniform depth of 4 to 5 mm and allow it to solidify. Preparation of inoculum. A. Using a sterile inoculating loop, pick a single colony of the test organism and inoculate it into a suitable broth, e.g., nutrient broth. B. Incubate the broth. Culture at 37 degrees Celsius for 18 to 24 hours to achieve a turbidity equivalent to 0.5 McFarland standard. Approximately 1 to 2 x 10 to the power of 8 CFU per milliliter. Inoculation of plates. Dip a sterile swab into the standardized bacterial suspension. Remove excess fluid by pressing the swab against the side of the tube. Evenly spread the bacterial suspension over the entire surface of the solidified agar plate by streaking in 3 directions to ensure a uniform lawn of bacterial growth. Allow the plate to dry for 5 to 10 minutes with the lid closed. Preparation of antibiotic solutions. Prepare serial dilutions of the standard antibiotic and the test antibiotic in sterile saline or broth. Typically, prepare dilutions ranging from 1 microgram per milliliter to 100 micrograms per milliliter depending on the antibiotic's potency. Cutting wells. A. Using a sterile cork borer or metal cup borer, punch uniform wells, 6 to 8 mm in diameter, into the agar at equal distances apart. B. Remove the agar plugs carefully using sterile forceps. Loading antibiotic solutions. A. Label the wells for different concentrations of the standard and test antibiotics. B. Prepare to fixed volume, e.g., 50 to 100 microliters, of each antibiotic dilution into the respective wells. C. Ensure no overflow occurs and the solution remains in the wells. Next is incubation. A. Allow the plates to stand at room temperature for 1 to 2 hours to let the antibiotic diffuse into the agar. B. Incubate the plates in an inverted position at 37 degrees Celsius for 18 to 24 hours. Measurement of inhibition zones. A. After incubation, measure the diameter of the clear zones of inhibition around each well using a ruler or vernier caliper. B. Record the diameters in millimeters. Calculation of potency. A. Plot the diameters of the inhibition zones against the logarithm of the concentrations of the standard antibiotic to create a standard curve. B. Use the standard curve to determine the concentration of the test antibiotic by comparing the inhibition zone diameters. Next is disc plate method. The method consists of placing paper discs saturated with antimicrobial agents on a lawn of bacteria seeded on the surface of an agar medium, incubating the plate overnight, and measuring the presence or absence of a zone of inhibition around the discs. In this method, different concentrations of antibiotics are used. After incubation, the zone of inhibition was measured and standard graph was plotted on graph by taking log concentration on x-axis and inhibition zone on y-axis. Procedure. Prepare sterile Na plates and with the help of a cotton swab spread the broth solution containing the test microorganism uniformly. Then, with the help of sterile forceps dip the sterile disc of filter paper into the different concentration of antibiotic solutions. Place the disc containing antibiotic on the surface of agar media. Incubate the plates in bod at 37 degrees Celsius for 48 hours. Calculate the zone of inhibition formed. Observation. In both methods, maximum zone of inhibition was observed at concentration 20 mg per milliliter of antibiotic. This content of slide, you have to write on left side of the practical file, if you are seen this video to prepare your practical file. I request to you, please subscribe this channel and like and share this video, to get more videos and updates. Thank you.